I'd like to welcome everybody to the June 13th meeting of GLIMPRINT and the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. Appreciate you coming today. We have a single talk this afternoon. As always, I need to remind you that these talks are live streamed on YouTube and recorded and made publicly available for later viewing in edited form. As always, Tom Helikar, myself, Jim Sluka, and Lorenzo Vischini are available to collect your ideas and suggestions for improvements. We have a variety of communication mechanisms, some of which work more than others. There's a LinkedIn page, there's the IMAG wiki page, there's the GLIMPRINT web page, which could use some love. So if people feel like contributing to that, we would be most grateful. There's the YouTube playlist of all the more than 120 seminars that we've had so far. So please help us distribute that. The web page has been updated. Jim Sluka has been working hard on that. It could use more content and more help. If people are willing to do that, we would be most grateful. We have a handle MSM Viral on Twitter/X. If you tag things that are appropriate with that handle, we will try to redistribute them, or you could use the GLIMPRINT handle as well. On June 17th, there's going to be a National Academies webinar on assessing the reliability of complex dynamic modeling and simulation that may be of interest to people. And so I'd encourage people to register for that if they are available. It sounds pretty interesting, and it's an important topic if we want to take these kinds of models and make them into things that are actionable. Reinhard Labenbacher is still hiring in Florida. People, of course, are very familiar with his work, and there are multi-level faculty positions in his Computational Systems Medicine Institute. And so please follow up with him if you're interested or know somebody who's interested in a position of that kind. We will be having our annual CompuCell training workshop beginning at end of July, beginning of August. If you can help us spread the word on that and get people to sign up, we would love to have more people. If you're interested in multicellular modeling, agent-based modeling, students, postdocs, anybody welcome to attend. There's also going to be a network modeling workshop. Herbert Sauro project, which is, of course, long-standing one that we usually coordinate with on the CompuCell side, is going to be July 22nd to 26th. In principle, deadline for applications is June 28th. It's a great way of learning this if you haven't done this kind of modeling using tellurium and antimony. And I encourage people to participate in that as well. If you've got a publication to share that's related to this topic, especially if it came out of something through this working group or GLIMPRINT, please do send it to us and we'll put it up to make sure that it is publicized on the GLIMPRINT page and the IMAG wiki. We will have a steering committee meeting tomorrow at 11 a.m. So Eastern Daylight Time. So if you are able to attend, that would be wonderful. If you think you should be invited and you haven't received an invitation, email me or Jim Sluka, and we'll make sure that you have the invite for tomorrow. Coming up, we have Paul Roberts, who's going to be talking about modeling of the eye to understand sight loss. John Metzgar has been rescheduled to talking about translating and applying Boolean network control theory to do drug discovery in cancer contexts. Pappalardo is going to talk to us about his whole immune modeling package. And then we have another talk on modeling coming up on August 8th. As always, if you have ideas about speakers, You're free to suggest yourself. You're free to suggest your students or your postdocs or the people you'd like to hear from. We would be delighted to get your ideas, and we'll do our best to get them on the schedule as we work forward. We would be most grateful for your ideas, especially a little bit of lateral thinking. It could be experimentalists, could be clinicians, could be people in drug development or biotechnology. It doesn't have to be academics. And if you've already spoken and it's been a while, like a year, and you've got new results, please feel free to suggest yourself for another talk as well. So I, as you know, don't do elaborate introductions. I don't want to take any more time from our speaker today. I'm very pleased to turn the floor over to him for an exciting talk, marrying machine learning and deterministic modeling of longitudinal single-cell data.
So thank you very much indeed for having me. I mean, it's a great honor to speak in front of this distinguished audience, and with all the things that have been introduced just now, I feel imposter, I must say. So I'm a biologist by training, who early on stumbled into the beauty of mathematics in a curiosity-driven way, and as such, I have a perspective on the field of mathematical modeling in biomedical research that some of you may find enriching, some of you may find controversial, but I'm happy to also discuss this. So as an occasion, I have a wedding here. We want to, because machine learning and deterministic modeling are getting married today, okay? And the context is longitudinal, so time-resolved single-cell data. And in essence, the wedding ceremony will have 4 chapters, okay? First of all, I will talk a bit about myself, just that you understand sort of my approach to computational modeling. Then I will elaborate a bit about the project, which I think was the reason why I got invited here, because I was awarded the Biomodels Model of the Year award for our work in the field of blood generation, so cellular modeling, molecular modeling. And then I will switch the layer from the molecular level to the more cellular level in terms of multi-scale, and speak about something that is just, it will come out hot off the press. It's about immune reactions in the gastrointestinal tract. And in the end, I think what will serve as a primer for our discussion is really how I see this sort of intersection between machine learning and deterministic modeling, so statistical and deterministic modeling, how this will develop in the context of biomedical research, okay? Because this is my sort of background. All right, so let's get started with a bit about myself. So for 3 years now, I'm a PI. I hope you appreciate the pun. In Hamburg, in Germany, at the University Medical Center. Again, I'm not a clinician by training. I'm a biologist who quickly turned into systems biology under the supervision of Ursula Klingmüller and Thomas Höfer. So I got the hardcore biochemistry and the hardcore sort of biophysics training. And I did my PhD at the German Cancer Research Center. And after that, I went for a postdoc to the Weizmann Institute in Rehovot in Israel, together with Ido Amit and Eran Elinaf. So Ido Amit is like who has an almost like engineering approach to single-cell genomics, and Eran Elinaf is a gastroenterologist, so a physician. So again, I had this really interdisciplinary sort of upbringing, which really sort of shaped my entire education. And I feel very privileged, and I'm very grateful that I have these wonderful mentors. And this all led to the fact that now, you know, there's on the one side, you have the wet lab where we do a lot of experiments and generate data. And on the other hand, you have the dry lab where we do a lot of theoretical considerations. And on a piece of paper, we start to scribble down some equations, stuff like this. And my lab is basically at the intersection. So I hope that the intersection is not an empty set, because this is where my lab is. And throughout the years, we were acknowledged in our work basically from both sides. So in 2017, I was invited to the Chaos Communication Congress, one of Europe's biggest computer hacking events to deliver a keynote there on how to hack the human microbiome. In 2020, I was invited to the DEF CON in Las Vegas with a talk. Unfortunately, it was only remotely, so I couldn't go there in person, but still, we got some recognition from sort of the computational stuff that we are doing. And also from the experiments, I was very delighted to got shortlisted and invited to the Lindau Open Science Days in 2020, and then last summer also to the Lindau Nobel Laureate meeting to the Lake Bodensee in Germany, where I could present my work in front of many distinguished Nobel laureates. And this kind of work was appreciated from multiple angles. So I received very early on already some seed investment from industry, which continues in other forms until today. I received some Young Investigators Award for my PhD thesis, which also laid the foundation for the work I will present later on. I received a Marie Curie fellowship from the European Union for my postdoc. And lately, I was awarded the Biomodels Model of the Year award for one of the mathematical models from the European Bioinformatics Institute. So you see that apparently there are some incentives for this sort of interdisciplinary approach. And to foster some more motivation, I would like to introduce the spirit animal, okay, for this kind of approach, which is the European green toad. Okay, so it's an amphibian animal. It lives in the wet and the dry, and similarly, you know, I feel very at home at the wet lab and the dry lab, and that's why this I think serves as a spirit animal. Also because you see the male animal here carrying the female animal from one point to another. So I think this is also a good spirit how we should in science support one another, and particularly women in STEM, I think. So this is the spirit animal, the European green toad. All right. I think in front of this audience, I don't need to spend so much on, you know, the conceptual foundation of what I'm going to talk about. I just would like to reinforce, you know, that we have these two sides of the same coin when we think of computational modeling at multiple scales in biology. On one side, you know, we have this top-down modeling approach, which is statistical modeling. You know, it's very static. We have very complex data sets we don't really understand. We can do some, you know, dimensionality reduction or more sophisticated regression modeling, machine learning, deep learning stuff. Then we don't understand, but we can still answer questions. We can stratify patients, for instance, and stuff like this. But for this, we need a lot of sort of data. It's a data-driven approach. So one classical example from the field where I work on is, of course, single-cell mRNA sequencing technologies, where with one experiment, you can, from tens of thousands of cells, for every cell, you can measure up to even 10,000 features, which are individual genes for which you can get transcript counts. And this gives you, of course, a lot of sort of data with one dedicated experiment. There's commercial vendors for all these technologies, so they are quite feasible. And therefore, it's very suitable for these kind of modeling approaches. And then they are sort of complemented by the sort of bottom-up approach, where we have more sort of dynamic data. And by dynamic, I mean time-resolved or longitudinal, right? This is also from the title of my talk. And there we have, it's a knowledge-driven approach, which means that we just look at sub-aspects. So if you look at the cell, for instance, maybe you only look at one molecular pathway that you're interested in, but you know already a lot about the pathway, and then you need high-quality data to even add on top of this, and maybe time-resolved data to understand the dynamics of the system. And then once you understand one part in a cell or a population of individuals, be it mice or men, right, you can start to sort of plug them together. And then from the bottom, you reach at some point the top. And classical sort of data set that is suitable for these kind of bottom-up modeling approaches, deterministic modeling approaches, you call it, are of course some sort of immune responses to infection that you have. So you have a cohort and you suddenly have an infection there, right? And then you sort of follow them during the course of the disease or during treatment, and you have different data points where you have very good understanding of the virus and its target cells maybe, and then you try to even better understand this mechanistically. And there you already see that maybe in these cohorts during the COVID-19 pandemics, you started to have even longitudinal data sets where you had from the blood single cell sequencing information. So both ends are sort of coming together right now, right? We are living in an era where actually these are not separated entities any longer, but we can actually marry them, right? And this is what's happening in my lab. And this is also sort of the outline for the talk. I would like to see that both sort of approaches sort of come together to take the best of both worlds, right? And to really understand complex data sets that then can also inform us about how things actually work in biology. And again, I might be very naive, and I'm very happy to also discuss this in a very critical manner after my talk. But I would like to give you basically two examples where we started to sort of approach and explore this intersection between statistical modeling on the one side and deterministic modeling on the other side. So let's get started with the project for which I was awarded the Biomodels Model of the Year award, how to handle bloody heterogeneity. And in order to start this, first of all, I would like to ask you all to take a deep sigh of breath. And to do this, what is required is actually that you have a lot of red blood cells that are populating your veins and your arteries, because they carry the oxygen that you were just breathing in to all the organs. And the moment I'm saying this here to you, or in order to do this and for you not to suffocate, it is very important that not the red blood cells, but their immediate progenitors, they are called colony-forming unit erythroid cells, or CFUE cells in short, they have to make a very fundamental decision. They have to decide whether they want to be or not to be, okay? Because they have basically two options. And this decision they have to make per second about 3 million times in an adult, right? 3 million red blood cells are generated every second that we are spending in my talk here. So 3 million times a second, these progenitor cells have to make the decision, do they want to become a mature red blood cell and proliferate and differentiate, or do they want to die? Okay, so it's a very fundamental process in a sense. And for me as a biologist, this was always very intriguing. So these progenitor cells, they proliferate, they divide so quickly. But then what we understand as cancer is basically if cells lose control over their proliferation capacity, right? So they are transforming and becoming cancer cells if they suddenly divide without slowing down, and they just lose all the brakes and they just divide in an uncontrolled manner. But these red blood cells, so blood cancer, leukemia, affecting the red blood cells is very rare. So despite the fact that they proliferate so quickly, they somehow maintain control. And I wondered, how do those cells do this, right? And the motivation for me to trying to answer this question was like a serendipity moment. I had this sort of, when I was a master's student, I think still in the lab of Ursula Klingmüller, I made this one like very simple experiment that gave me a very puzzling result at the first glance. So let us look at this result. So what I did is basically I prepared these progenitor cells of the red blood cells from mice, so from mouse embryos to be precise, from the livers of mouse embryos. And then I treated them in the lab with this hormone, erythropoietin, which is the only factor they need to survive. So if they have this hormone, they just live happily and they divide quickly, as I said. So I just, threw some EPO on them in the lab, and then I stained sort of their intracellular signaling to sort of follow them, how they get activated, and follow the molecular machinery that they need in order to survive in the dish, right? And then one of the key factors for the signaling is the transcription factor called STAT5. And STAT5, in order to be active, STAT5 needs to be phosphorylated. So you have this phosphostat5, this pSTAT5 here. And then I just stained with an antibody, I stained the phosphostat5 molecules inside of these cells, and I observed this violet distribution here. And this was unexpected because it was so broad. Okay, and then I thought, maybe I did something wrong. Maybe the antibody was not correctly calibrated, or the antibody is not a good antibody, and the fluorophore with which it is connected, it doesn't really work. But I did all these controls, and I always had this broad distribution. And I wondered, how come this cell can afford such a broad distribution of STAT5, which means that some cells have so much more STAT5 than others, if this is the only factor that is really crucial for their single important decision, which is to die or not to die, right? So what's the biological reasoning about this? How come a cell can afford this? So to answer this question, we needed some mathematics. So let me do some interlude on how we sort of model this. So as I said, we looked at this sort of STAT5, which is a transcription factor, which means it binds to the DNA in the nucleus of these cells, right? And now we basically wrote down ordinary differential equations, which basically describe how the abundance of this transcription factor and all the signaling molecules that come above and also that come afterwards, how they change over time, right? And this, together with the data that we measured, we can basically put into the computer, and we basically write down this model as a set of coupled ordinary differential equations. And not only do we have an ODE for the mean of the population, so how an average cell sort of activates STAT5 in the nucleus, but also we had, you can think of this as a second moment equation, you also have an ODE for the variance of this signal within a population of cells, right? So we can take into account this broad distribution of STAT5 that I measured in this first experiment. And then, it's just a matter of how you implement the routine to do the optimization, the fitting, the parameter estimation. We basically did this. It's all published. You can read up in the paper, but I would like to spend a bit more time on the conceptual approach, why we chose this. Because once we had this sort of written down and in the computer, we could basically ask some questions. For instance, we could ask, what are the causes and the consequences of this bloody heterogeneity in STAT5, right? And then the model said, give me some time. But no, of course, we optimized this process. So we got an answer in a reasonable time. So basically what we did is we looked at sort of cross correlation and asked, okay, the variance in phosphostat5 between cells is so high, which other parameters or variables also have a high variance? And then the model suggested that one reason for this high variability in STAT5 is actually import and export rates of this transcription factor in and out of the nucleus. I told you that STAT5 needs to be in the nucleus, right? And so it said that, you have, for instance, a broad distribution also of this import rate from one cell to another. Sometimes the transcription factor gets imported into the nucleus very fast and sometimes very slow. And this was, of course, interesting, but we wanted to experimentally validate this kind of prediction. But it's very hard to follow individual molecules and individual cells and then make this calculation whether we see this broad distribution. So we came more from a physics approach and argued, okay, but these transport processes are some sort of directed diffusion, right? So they should indirectly scale with the volume of the compartments, right? So if you have a very big cytoplasm, it will take an average molecule very long to just go to the nucleus. However, if the cytoplasm is very small and the nucleus is very big, just by chance, the transcription factor will move into the nucleus very quickly. And so under this assumption, we said, okay, so we can also just measure the distribution of the sizes of these compartments, right? And when we did this sort of microscopy experiment, we found that sort of the prediction of the model was sort of in line, that we have a very broad distribution of the cytoplasmic sort of volume, which again is sort of proportional, particularly when it comes to the variance to these import rates, right? So we found this model prediction to be validated experimentally, and since we had quantitative data, so we were able to really count the molecules of STAT5, we could now also calculate with some downstream signaling that we measured and also measuring how many cells were proliferating and dying, how many molecules were actually required in the nucleus of these cells in order to make these threshold for surviving, right? So we measured certain apoptosis markers, for instance, and then you could tell, okay, some cells do always survive and some cells do not. And it's a property not only of the amount of STAT5 that they have, but also the volume of the compartments. And so we figured out that actually it's sufficient for a cell to have in the order between 20 and 100 molecules in the nucleus in order to survive, which is very interesting. And again, think about our original observation. broad distribution of STAT5. It seems like the cell does this to ensure that you always have a minimum number of these cells that certainly survive and really replenish all our red blood cells so we won't suffocate, okay? So I think here, this noise that we have is actually not bad. It's not a loss of information, but it's a way how the cell makes sure that you always have some cells that survive and sort of replenish red blood cells for us not to suffocate. So this was very nice. Of course, we asked, is this something that is applicable beyond red blood cells that are, of course, important in case of, blood loss or wound healing or something? And we found that also other members of the STAT transcription factor family, for instance, STAT1, in response to interferon gamma in these cells, shows a very similar behavior. So this seems to be something that, at least for this family of transcription factors, seems something that is not only related to a response to erythropoietin, but also maybe to some more immunologically relevant questions that are of interest for you guys. So with this, we also made it to the cover of Cell Reports. And I think it, just laid the foundation that we ask ourselves, okay, not only what does an average cell do, but also what does an individual cell do, right? As a representative of a population that might not well be represented by the mean. Okay, so, but then sort of, this was the work that basically originated from my PhD, and then I was a postdoc, and then I started my own lab, but this idea was still revolving in my head. But now I changed sort of the environment, and I went to a university medical center, right? And there I started, read about a lot of stuff. And here I'm in the first department of medicine that is internal medicine. So I came across inflammatory bowel disease, IBD, where patients basically suffer from cramps, from diarrhea, from weight loss. And if you think you have nothing to do with this, even though for you who might be in, immune related research, no matter where you are in the world, whether you're in the US or in Europe or even in the East, in Asia, it is anticipated that by 2030, which is pretty soon, the incidence rate of inflammatory bowel disease will reach 1%, which is 1 person in 100, okay, who will be affected by this disease. And not only the incidence rates are skyrocketing, but also some technology, okay, which is single cell sequencing. So there is more and more of this kind of data available also in the context of IBD. So some people already saying, we are entering a new era in biology, the era of big data. But in my humble opinion, there's only one big data. It's him. But if you look at big data from the right angle, you can even already discover some AI here, right? So artificial intelligence. But for me, as a biologist, who stumbled in this computational modeling, sometimes I feel a bit overwhelmed with all of this. So here I depicted myself with the help of a generative artificial intelligence. So you see me and my sort of mathematical model, and now I try to fit everything in there. So I have one type of data, which is flow cytometry. Then I have single cell RNA sequencing data. I have some sort of in vitro validation experiments. And then I have also a human cohort. And I try to sort of fit it all in. And this is sometimes not easy, at least for me, it's not easy. That's why I want to share it here with you. So I would like in the last 10 minutes or so, I would like to show you one project where we try to, fit it all in. I started, reading in the field of IBD, and I stumbled across, again, a very simple experiment where we had single cell data, so flow cytometry, the same sort of experimental setup that you have seen in the first project, of a mouse model of IBD, where at different time points during the mice who get first sick, their gut gets inflamed, and then it's starting healing when they are switching from the DSS to the drinking water again. The cells from the intestines were isolated and basically measured by flow cytometry. So we ask ourselves, how does IBD look like to a computational biologist or to a systems biologist? And we have this DSS, which basically induces some damage in the gut, and then the gut is also healing, but it also has an effect on the turnover rates of these different immune cells that you have there. Okay, and all of this was measured. And then, of course, we simulate the presence or absence of DSS, but then we ask, okay, can such a very simple sort of model describe how the abundance of immune cells is changing over time? Immune cells that were total numbers were counted, okay, per time point. And the answer is yes. You get a qualitatively good agreement, and also we provide all the metrics to evaluate the goodness of the fit. But then we ask again, can we generalize this? So we looked at a different paper and a different data set where they also looked at a mouse model of IBD, but now a different concentration and a different timing of DSS and also different technology. Here they performed single cell RNA sequencing. So at different time points, they killed the mice and they looked at the single cell sort of abundance in the gut of these mice. So first of all, we had to make sure that we look at the same sort of cell types that we were looking at in the first experiment, right? So we reclustered the data. So this is the machine learning part, right? And we sort of annotated the cell types that people looked at in the first experiment. And also now, in addition to the immune cells, we also had the epithelial cells that are lining the inner mucosa of the gut, okay? And we made sure that all the sort of surface markers that they found, that they used in the first flow cytometry experiment, that we also find them in these populations that we annotated here. So this looks sort of kosher. So we sort of went on and used the same mathematical model, just extended to this epithelial cells. And the very same mathematical model was also able to describe sort of this, again, different protocol of a mouse model of IBD. So we were very pleased to see this, but of course now we ask, okay, what can we actually learn from this? Now we sort of understand how are the immune dynamics in this inflammatory disease, but is there something emerging from this? So actually we looked at all these different turnover rates that were estimated, okay? So they were all identifiable, which means that we had sort of narrow 95% confidence intervals of these parameters. And when we compared them for the two different data sets, we discovered a certain hierarchy here. And we said, these are turnover rates. So biologically, what they mean is that cells with a high turnover rate, either they die slowly, right? Or they proliferate fast. I mean, we cannot discriminate the two because we merge it all into one. But so we set out to perform a dedicated experiment to sort of validate this. So we also did some sort of intermediate protocol of the IBD mouse model. And then we stained for Ki67, which is an antigen, a protein that basically is upregulated when cells start to proliferate. And when we did this experiment, we found sort of this very same hierarchy that was also suggested by these estimated cellular turnover rates. So this gave us some confidence that these estimated turnover rates are actually meaningful to describe the dynamics of immune cells during inflammatory bowel disease in mice. But now we moved on to human. So I read this article about an open label trial of IBD patients who do not respond to any treatment. Okay, so these people are very, it's very unfortunate that, whatever they were given, nothing really worked. And maybe this is the reason why they agreed to be part of this trial, where while receiving this new drug, they underwent 6 times intestinal biopsies. So for six times during a course of 14 weeks, they got a biopsy in their gut, and then the cells from the gut were sequenced, but not on single cell level, but just on bulk level. So the first thing, in order to make it comparable to our previous approach, we had to deconvolute it, right? So we looked at which fractions, given the transcriptome, we expect these different cell types to be present. So then we used sort of the same model again that was just semantically adapted a bit to the human context. So here, of course, we don't have DSS, but we just simulated some input cue that sort of triggered the disease. We have some active disease, and then we have also some resolution. And we looked at this deconvoluted data now from the bulk sequencing, but we focused on the few patients who actually entered remission, because in this study, they observed that some people entered remission, which means that the symptoms went down and didn't come back. But actually, the authors didn't really understand why this was the case. So we thought maybe our model can help to understand this. So the question is, of course, can this model sort of describe these deconvoluted dynamics? And the answer is, again, yes. So it seems like the model can describe the dynamics, and now we can use it basically to test some more hypotheses on why these patients basically entered remission. So one hypothesis the authors had was that there's an increased neutrophil abundance, which basically is there in the first place. But if you have many neutrophils there, then these people somehow are more susceptible to respond to the drug. But then when they measured myeloperoxidase, an enzyme that is really highly expressed by neutrophils, they didn't find a good correlation. However, when we now basically looked at the correlation between our sort of simulated cell abundance from the model and what they measured of phosphorylated STAT3 in this case, which is a direct indicator of the drug, which is an IL-6 inhibitor. So they measured this in the upper layer at the gut epithelium, but also in the lamina propria right below. And we found that indeed only the neutrophils are positively and significantly correlating with this. So it seems like that if we use the model, then maybe we can, with the simulated amount of neutrophils, we get an indication who is responding to the treatment or not. So then we thought, again, maybe this is a more sort of general observation. So we looked also in other data for other treatments of IBD. And also there, for anti-TNF treatment, infliximab, the drug is called, among all the responders, classical neutrophil markers were upregulated before treatment, which again indicates that the high presence of neutrophils in the beginning might be indicative of treatment response. So this is a wonderful team effort by actually a clinician scientist, a digital clinician scientist, and a more sort of biologist who now is also turning into a computational biologist from the lab. And as I said, it was just accepted to NPJ Systems Biology and Applications. I don't have the proofs yet. I cannot provide the link. I can only provide the link to the GitHub where you can already find all the process data, the code, so you can rerun the model, try it. So again, I really would like to acknowledge this interdisciplinary approach. Which brings us for the last minute or two to a sort of outlook, how I now see this coming together of the deterministic and the statistical modeling in the future. So what I basically envision is that, in the clinics, it's not realistic to ask for, clinical trials where you have longitudinal sampling of patients and at every time point you do single cell RNA sequencing. But maybe we can have a combination of both. And of course, I think for you, the audience, it's maybe think of a viral a cohort with a viral infection. So we also have a project on people living with HIV. And there we could, at the beginning, before treatment, maybe we do a deep molecular and cellular profiling by single cell RNA sequencing, but then we only do a very straightforward flow cytometry experiment. So we do all the machine learning, and maybe also we parameterize our deterministic model based on what we see in the single cell data. And then the mechanism comes when we meet the longitudinal information just based on cellular abundance. So this is sort of how I envision this. And then, this mathematical expression basically means that if A is true, then also B is true. So I think, if we are very good at really understanding the single cell patterns that we can measure, then we will also be able to marry this with our very quantitative and dynamic understanding of what happens in these populations of cells as an immune response, right? So to sum it all up, I've shown you that machine learning and ordinary differential equations can be married for longitudinal single cell data. Ki67 staining seems to be particularly indicative of cell type specific turnover rates. And we are applying this now, and we have very exciting work in progress, I must say, in metastasis formation, and in this context, also viral infections. So we have influenza infections and CMV infections. So maybe, I will come back to you in a year from now to also present you this. But since we also have quite some time for discussion, I wanted, of course, you can ask whatever you want, but I had like 3 primers for discussion. So I really wonder from your experience also, what are your bottlenecks in fair modeling? And fair means findable, accessible, interoperable, and reusable. How can we basically increase visibility for us who are, computationally tackling this, and to make people aware of, chances as well as risks of these kind of approaches, limitations, because we don't want to sugarcoat anything, or we don't want to, sell it to people in a sense, but we want to use it to answer questions, right? And then, I mean, now that I have this have seen this introduction, where can we actually connect to really grow as a community? And maybe, we also want to write some more opinion pieces from our perspectives. I think this could be something that really is important. So this leaves me with just appreciating the people who did the work, my wonderful group, and of course you for joining here. And I'm happy to take questions.
Okay, thank you for a great talk. The floor is open for questions. People should be able to just unmute and ask questions, but if you can't unmute for some reason, you can put it in the chat and we'll make sure that you're able to ask your questions.
Hey, Lorenz, that was an excellent talk. Very interesting. A quick question. Your pStat5 constant count or measurement and your cell volume measurements don't seem to be on the same scales. You've got orders of magnitude variability in pStat, but you've only got differences in cell volume of a factor of 2 or 3. So it doesn't really seem like, basically what the argument is that, well, you need to go by concentration and not amount in the cell. Concentration is what drives the chemical reaction, not molecule count numbers. But those two figures seem to be disconnected by one on a log scale and one on a linear scale.
Yes, that's a good point. Does it help if I jump back to the slide?
Yeah, sure, that'd be great.
Yeah. So
I mean, certainly it would get the trend in the right direction. So your volume is changing by a factor of 8, from tail to tail.
Right.
But your probability density is changing tail to tail 3 logs. At least, if not more like 5 logs. Depends on whether you're looking at the purple or the blue curve.
Yeah. I see your point. So you have a suggestion, like what does this suggest to you? There's multiple effects, right? That probably potentiate one another. So in addition to having this directed diffusion, you must have actually, and it's also correct because, as I say, these import rates, they are one factor that contribute to the variability, but of course they're not the only factor. So I think I even have a backup slide where I show how these individual aspects are basically contributing here to the sort of covariance. So you have, for instance, the receptor for this growth hormone erythropoietin, where you also have some sort of broad distribution, however, not in the total amount of STAT5. So you have not a lot of fluctuations in the total amount of molecules that you have present in a cell, from which a fraction only gets phosphorylated and then transferred to the nucleus. And then again, you have a broad distribution for these import and export rates. So yeah, they contribute these rates, but they are not sufficient to explain the whole range of variability. So very well spotted.
So an alternative explanation would be that the system is saturated.
Yes.
That you have more pStat5 than you really need. So whether pStat5 is 10 to the 5th or 10 to the 6th doesn't really make too much difference because 10 to the 3rd saturates the available binding site. Wild ass guess, you might say, well, how many pStat5 sites are there in the human genome? And, it's a regulatory factor. So figure there's 100 plus or minus a factor of 10. So a cell probably needs like 100, a couple of hundred transcription factors in the nucleus to activate all of the genes in the nucleus that respond to that transcription factor. And it seems to me like your numbers for counts in the nucleus or even counts in the cell are above what you would think you would need, suggesting that that pathway is saturating. And so factors of 2 or 3 in pStat just don't make too much difference. But the whole system is complicated by the fact that the binding constant for every single nuclear receptor site is different.
Yeah. You have different target genes. And also what's even more complicated, there's 2 versions of STAT5, A and B. and they can make homodimers A and A, or B and B, or heterodimers A and B. And it has been shown by chromatin immunoprecipitation that depending on which ratio of A and B you have, different genes are bound.
Right.
So yeah, so it's of course
it might be
more
also
complicated
interesting to look at just the expression levels of the other transcription factors and nuclear receptors. Because the other thing that happens at a transcription initiation event is there's half a dozen proteins bound to that gene. And you know, STAT is a heterodimer, but there's so many other proteins sitting on that gene that there may be, you know, crosstalk with other transcription factors. You know, some genes require absolutely 2 transcription factors to be bound, 2 or more. Just one will never turn the gene on. So you get in a very sticky matter of, well, there's at least 100 binding sites, and oh, by the way, every one of those 100 binding sites has different local transcription factor requirements, and you know, just too many things going on at once.
Okay, I think what this would require even like some sort of Boolean model for, you know, binding of the different target sites downstream of STAT5. And here, I must say this: we only focus on phosphostat5 because you must know that we calculated the total amount of phosphorylated STAT5 molecules by targeted mass spectrometry, which means that we had a very specific peptide, a very short stretch of amino acid that gets phosphorylated in the STAT5 as a sort of synthetic peptide that we absolutely quantified, and then we always spiked in the measurement as a sort of ruler to calculate then the native signal and calculate back how many molecules of phosphostat5 we had. So this is not straightforwardly scalable to many other transcription factors. But I would love to do this, obviously. Yeah.
I have a related question. So this is Chang from Emory. Nice talk here. So the STAT5 fluorescence, flow cytometry fluorescence intensity there, right? So that represents the concentration in the nucleus, or actually represent the total abundance of these molecules in the nucleus?
So thanks for the question. In essence, in the paper, we did all possible combinations, because what you can do is you can, I mean, with flow cytometry, you cannot, there you only get the total amount. No matter if it's in the nucleus or in the cytoplasm, you can basically not discriminate unless you have a way to fix and permeabilize the cells that only the cell membrane gets sort of leaky, but not the nuclear membrane, which is tricky. So for the flow cytometry, we only had the total amount of phosphorylated STAT5, but there what we could do to quantify is to have beads that were quantified with the fluorophores. So then we had a total amount of fluorophores as beads that we spiked in, and we had as a reference population in the scatter plot. And then we could sort of calculate back to the fluorescence that we got from the phosphostat5 that is bound in the cell, given that the antibody was linked to one fluorophore at the time. So this was in a flow cytometry experiment. But then I told you we also had the mass spec experiment. And there we could, of course, biochemically in a very sort of clear way separate the cytoplasmic lysate from a nuclear lysate from a total cell lysate. And there we basically could look at all the free fractions separately and could see whether then the total number of STAT5 and phosphostat5, whether they were adding up. So this is what we did. Okay, so if you're able to normalize with the volume you measure, then that variability will not be as huge as you're seeing there, right? I mean, cross the fluorescence data crossing like a 4 or 5 order of magnitude. I'm just wondering. Yeah, exactly. So this relates back to the question that Jim asked, whether, I mean, what is it that carries biological information? Is it a concentration or is it an absolute amount of molecules? Right? And in this project, I think we saw that you cannot really disentangle this, because while the volume might affect how quickly you can bring molecules from A to B inside the cell, you need an absolute number of molecules in order to satisfy all the binding sites on the DNA in order to release a certain program of gene expression. So, and also this is, I think, particularly interesting because we know from some sort of viral response genes that you have a lot of target genes that are actually not changing on the mRNA level upon a viral infection, but their mRNA has a very long half-life and it's basically constantly produced in a very high level. But once you have the viral cue coming in, this mRNA is just translated. And of course, the protein that you then need in order to fight the virus is made quickly. But if you would only look at mRNA expression of target genes of some of these interferon response genes or key signaling factors in this pathway, you probably wouldn't even see a difference. So again, biology is so wonderfully complicated. So we have concentrations that factor in. We need absolute amounts of molecules, but then we also need the system to be in a sort of steady state that puts it into a situation to even respond. Because if you haven't this for whatever reason, because a cell is anyways a very filled up, busy, watery sphere. And then if your sort of steady state for the mRNA is sort of perturbed already, then also something that you might not directly connect to it might also be perturbed. So yeah, it's all sort of connected. And if I learned one thing, in biology, then there's hardly any situation where it's really black or white. So usually you have all these different shades of, sometimes it's more like that volumes are being factored in. Also, in terms of biology, you might wonder, why would a cell even change the size of a nucleus or the size of the cytoplasm from one generation to another? In these cells, progenitors of red blood cells, it's particularly important because red blood cells, they don't have a nucleus. So as they differentiate, the nucleus gets smaller and smaller, and at some point is thrown out of the cell. So when they are red blood cells, they are basically just bags of hemoglobin, and there's no protein synthesis done anymore, no transcription, nothing. But they are just sitting there then for 120 days in humans and 30 days in mice, and then they are being eaten up by macrophages or just dying, So this is, again, why on a biological level it makes sense to have this regulatory element to vary compartment sizes, but then also look at different molecules on transcription level, on translation level. So everything sort of makes sense. We are just starting to understand this. And I think all these models can really help us to discover these kind of mechanisms.
Thank you.
No, thank you.
Other questions? I may ask a follow-up. So you mentioned that as you point out, red blood cell cancers are rare. Mm-hmm. Although I did know somebody who died at age 30 from one, so it does happen.
Yeah.
So what are the key, if we came back to sort of the understanding of the control, what are the key dysregulations that occur when you do get a red blood cell cancer?
Yeah. So this is actually something that we looked at in another project, because unfortunately the JAK-STAT signaling axis is not the only one, and it's only important for ensuring anti-apoptotic signaling, so a cell to survive. But in order to proliferate these progenitor cells, the process that is mainly deregulated in the red cell leukemia, you need mostly MAP kinase signaling, and then also AKT, PI3K, AKT signaling. So in a different project, we basically looked at these two sort of signaling pathways, and then we asked the question, is it one pathway or the other that is sort of losing control, and then these cells proliferate out of control? And it turned out, so we also sort of established a mathematical model, and we sort of calibrated it with whole proteome measurements that we got from these sort of patients, because at some point they get some leukapheresis, which means that all their blood cells get mobilized, and you get, with one, you take one sample of blood and you find so many stem cells that you can then differentiate into these progenitor cells of the red blood cells, that you have a lot of biological material to study them and to make all these measurements. And long story short is, what we found is that for 3 patients or for 2 patients, you will never find the same sort of weights that are attributed to the different pathways, which mean, however, what we were able to do is we take a steady state measurement of these protein abundances. So anyways, this was a hypothesis there. Protein abundance is what matters, because these signaling pathways, those are enzymes, right? Kinases. And I think enzymes, they just do whatever they want. If they have enough substrate, they will just phosphorylate every substrate as fast as they can. So the rate limiting step is usually not the amount of phosphate that they need or ATP, and it's not the substrate, but it's just the velocity of the reaction just depends on the total amount of enzyme that you have, unless you have a mutation, of course. So we were able to just, to have a calibrated mathematical model. We measured with the global proteomics the abundance of all these signaling components. We plugged it in and we simulated how active are these different pathways and mapped this to the proliferation of the cells that we measured. And then, there was not a single patient who showed the same weightening of the two pathways. However, from this snapshot steady state measurement, we were able to computationally recommend either inhibiting one pathway by a MAP kinase inhibitor, or the other pathway by a AKT inhibitor, or a combination of both. So this was at least some way how we could rationalize this treatment based on this one snapshot proteome measurement that basically factors in how active are the PI3K AKT pathway and how active are the MAP kinase ERK pathway. So, but still, we are just scratching at the surface here as well. Yeah.
Great. Thank you. I mean, it comes back to something that is always a bit problematic in any of these models. It's not yours particularly. If you look at any of these nice disease pathway diagrams that you showed during the course of your talk, you'll have a molecular concentration, and then that will couple to a cell output, like cell death, rate of cell proliferation. But if the world had a simple relationship between the concentration of molecule X and those complex behaviors, life would be easier to analyze. On the other hand, from the point of view of mathematical modeling, we have to start with those hypotheses. But you showed a couple of very elegant disease maps, what I'd call a disease map, in the later parts of your talk, and I always wonder how. Ultimately, I suppose the degree to which your model fits the observations is the validation of those kinds of hypotheses. But it's always problematic for me when I look at omics data to try to couple them to the behaviors at the cell and system level that are the outputs you care about medically, at least.
You're absolutely right. I couldn't agree more. Unfortunately, I don't have a solution to this. So usually what we do, we have these calibrated models that can describe the molecular dynamics that we measure. And then we call these what you said, like proliferation or death. We call this emergent properties because, right, this is something that is coming out of the system. And the only way we can sort of link them is by regression modeling, which means that we sort of integrate a certain behavior. For instance, we look at how many STAT5 molecules are in the nucleus over time. And then we run an optimization and see, okay, it seems like for the cell, the first 45 minutes after stimulation are most decisive then in order to describe whether they undergo apoptosis or proliferation or something. So we link this by some sort of very simple linear regression, and we see sort of, we test all these different integration times and then find what is doing this link best. And then, of course, we can look at other conditions and see whether this regression still holds in order to predict from the molecular ensemble also the emergent property. But of course, this doesn't mean, for one, that we understand how this is then biologically really sort of implemented, right? I mean, I understand what an integral means in the sense that a transcription factor needs to sit on the DNA for a certain time in order to recruit the polymerase and drive gene expression. But apoptosis, for instance, is a very complicated process that is more than gene expression, right? You don't even see it on expression level so much. That's why we also looked at Ki67, because this is a protein, but having caspases that are active or cleaved or something is a whole different level of complexity. Something similar with phagocytosis or endocytosis, right? Many different processes on different layers of molecular entities happening at the same time. And all we do is, we do a linear regression and basically connect two dots with a line. So yeah, I think we need more ideas how to model this or how to interpret this then, and how to really first of all observe and measure a cell as it undergoes such decisions. But then also, again, there is already a lot of data, and I think currently the bottleneck is really that we are coming together and really think together about, how can we basically implement this? And I think you, with your initiatives of the, this virtual cell, CompuCell, this is going into the right direction. But you, or not you need, but I think there's so much we can all we should all contribute to this from our different perspectives. And yeah, I still wonder, so again, that's why I set this as a primer for the discussion, how we can further foster this. To also, as you said in the beginning, also involve clinicians, right? So when I introduced a clinician to the second project, his perspectives, because he's on the ward like every day and he talks to these patients. So for him, it means a lot if these patients do not respond to treatment, and he has a very different motivation to cure this disease, but also, how he approaches this phenomenologically. Mind models are also very important as a first step before we write down equations. So I think we need all of this, and we need to make people aware of how we think, how we see the world, and what we can contribute to this very big jigsaw puzzle that is basically how cells make decisions in health or disease situations. So yeah, I think you asked the right questions. I'm very sorry I don't have a perfect answer to it, but this is certainly something we should look after in the future.
Well, one thing that Jim commented and I were chatting a little bit on the side is the studies you can do are longitudinal in the patients, but not in the cells. Mm-hmm. So you can't follow in general. Certainly in the patient, you can't follow the time course of chemical concentration in a single cell.
Yeah.
And so our ability to do omics has gotten better and better and better, but that's always in fixed. It's always in fixed cells. You can't actually do a longitudinal study in a single cell. I mean, you could do it in vitro following a few channels. But you can't do all the beautiful mass spec that you can do now. You can't do the RNA-seq and so on. And so what we can do in a single cell longitudinally is still fairly limited. And I think that potentially makes the coupling of behavior to levels, which would be complex anyway, even harder to nail down. I'm not saying it's not a limitation of what you're doing. I think it's a statement about where we are experimentally in our ability to monitor the state of living things.
Absolutely. I couldn't agree more. And that's why we need both. We need the theoreticians that provide more frameworks. So for instance, now I've seen there was this preprint from the group of Uri Alon where they look in 2D into a tissue, and they have certain cell types that they identify, and they only have information about a few different proteins. And from those, Ki67 is one of them. And from those, they try to predict how these cells will sort of behave in a state space where you also have cell death as an option and proliferation as an option. And from this snapshot, now they try to, of course, in state space, you just can find an attractor and cells will certainly end up there going crazy, proliferating or dying. But of course, you don't know when they will reach it, right? So there's still conceptually things that are difficult, but many things that still can be tried. But then also we need the experimentalists who maybe provide us with new technologies. And again, we already have a lot of information, maybe not the right quality of information. I also see this limitation. We can't, live follow cells as they develop in vivo. I don't see this it won't be solved on short term. So yeah, until then, I think we have to make the best out of what we have. And I think the only solution here is really to bring people together from different angles, have events like this, and talk to one another. So thanks again for having me. I really enjoyed it. And also the discussion is, even though it's frustrating at times because I feel that we are all struggling with the complexity, it's also very enriching for me, at least.
Are there any other questions from the audience? I see some people who often ask questions here. I don't want to monopolize the floor. I have one or two others, if it's okay, but I want to make sure that others have their chance.
I have a question.
Go ahead, please.
Hi, Lorenz. This is Melania.
Hi there.
Sorry for my voice. I've been sick. So regarding the STAT5 project, so you mentioned that the cells kind of agree on having a huge variability for the reason that you very nicely explained. And as usual, here the questions also from my point of view, and I have the same question for many other types of cells. So the question is always, is the regulation at the level of the population or at the level of the single cell? So for example, if we look at a specific cell, will this cell change the quantity of STAT5 over time, or that cell has kind of always more or less the same amount of STAT5, and the regulation is achieved, the variability is achieved at the level of the population? And the follow-up question is also whether you can check this with single cell sequencing.
Yes, so thanks for the question. I can only answer it in this very specific context of STAT5. But in general, this can be answered easily by reporter mice. So basically mice where you have some sort of fluorophore that is basically connected to every STAT5 that is produced in a cell. And then you can basically, in terms of live cell microscopy, just follow this cell during the lifetime of the cell and see whether the total amount of STAT5, for instance, also in the nucleus, is changing, yes or no. And these experiments have been done with STAT5 reporters, and the answer is basically that this fluctuation is a property of the population. So it's not that an individual cell is going from one end of the distribution to the other end of the distribution throughout their lifetime, but it's really that you have an ensemble that is very heterogeneous in the first place, already at steady state. And then this might come in handy if you, for instance, have a situation of blood loss where you suddenly need many cells to proliferate. So you already have some of these cells in the population which have a lot of phospho-STAT5, so they are very prone to react here. However, whether this generalizes to other members of the STAT family or to other sort of immune situations, we've mentioned interferon alpha and interferon gamma responses, I don't know, but I think the literature is should come in handy there. So this is for reporter mice, so genetic. And then you said, can this be inferred from single cell sequencing data? So also there we are working on some data sets where we have these kind of reporter mice and we do single cell sequencing, and then we also find transcripts of the reporters, which means that we can sort of trace them back, and we sort of know based on the expression of a certain fluorophore whether they ever express some canonical markers during the course of a T-cell differentiation, for instance. But it's still tricky, mainly for, at least in our hands, or in the data I was working on, because some of these regulatory, the key regulatory factors in these T cells, they are very sparsely expressed. Like on the transcript level, you miss quite a lot of them, which means that if you have a cell where you don't have this fate marker, how can you basically know whether it has never been there or you just don't detect it anymore? And I'm not a big fan of imputation here. So usually what I like to do is I use the fluorophore, I use the flow cytometer, and I sort a very homogeneous population of all the cells that have this marker fluorescently expressed in one vial. And then I do bulk sequencing very deeply on those. And I compare it to all the ones who don't have it, which doesn't mean that I also have some heterogeneity within these two populations, but at least I can look at the bigger picture comparing one to the other. And I think for when we talk about immunology and antiviral response, it's still the majority of the population that will decide what happens. So if you have many cells in a population that can fight the virus, you will be able to constrain the infection. So a single cell will not be sufficient. However, in the context of cancer, this is completely different. There, a single cell can be a tumor-initiating cell, right? And if one sort of immune cell has the right receptor, then this clone can also be propagated and can solve the situation and can, you know, attack the tumor. So I think in terms of tumor biology and immunotherapy, their single cells might be well decisive. But in other contexts, at least my impression is that the majority of the population is always the one that is responsible for a certain phenotype of the entire organism.
Thank
Very
you.
good question. Yeah.
Thank you so much.
That was an interesting point. I mean, it's a lot easier to treat. I mean, for example, let's say genetic engineering. It's a lot easier to treat loss of function diseases than gain of function diseases, right? Your cancer, right? You have one cell that misbehaves. Cancer is an example. You worked in polycystic kidney disease, which is the same thing. You have a single cell with that adverse phenotype. It can take over the whole kidney. So then the treatment has to be 100% effective because you have to get rid of all of the cells of that type. Whereas if you have to supplement something, then it's an easier task. So I think that's a great point that you just made.
Yeah.
Other questions? I wanted to ask you a question. You put up on your slides, just to begin with, something we haven't talked about, which was fair distribution. And you mentioned already what you called a mental model. I would typically call conceptual model. And you showed some diagrams. I don't know if you want to come back, maybe pick one of the examples where you had what I'd call a disease map, maybe for irritable bowel or one of those other slides. We could look at that together if you're willing. That one would be fine. And so here, you have a diagram, and which embodies the conceptual, what I call the conceptual model, although it also has rates on it, so it has a mathematical version of it. We could do that when they're networks. We have a lot more trouble when we have spatial interactions. If you wanted to say, show two cells are synapsing with each other to activate a T cell, for example, we don't have a visual language that's as well developed to describe those things in a systematic way. And so I think that's something that might be an issue that's worth thinking about. Maybe in terms of blood, it's a little bit less of an issue, but in a lot of other diseases, spatial relationships between cells are important. And
absolutely.
So that's something I thought maybe would be something you might want to talk about a little bit in terms of the way you're thinking about that kind of problem. How do you actually get that conceptual information?
Thanks for that question or remark. So we are working also on spatial transcriptomics of the gut in case of IBD. And there, what I find particularly interesting is, of course, there also you can look at gene expression modules. So, but you don't get down to the resolution of a single cell, but you have resolution of single spots, so where you have 2 to 10 cells. Okay, and then what people do is they look at these gene expression modules at the spot resolution of 2 to 10 cells in the gut, and they sort of connect it to single cell gene expression from the same condition. Sometimes it's from the same mouse, usually it's from another mouse that is from the same cohort or human being. And then they try to sort of map this. However, it's mathematically not precisely doable that you really do a deconvolution in the sense that you say, okay, in this spot I have 20% of T cells and 80% of epithelial cells or something. It doesn't really work. But I think it's for the reason I've mentioned before, it's not so important, because if you have a very strong, let's say, pro-inflammatory, like a cytokine profile in this spot, it doesn't really matter whether it's coming from 20% T cells or 80% T cells, it will make the damage regardless. So there, it's not a problem. But one thing that I found that I started thinking about, and I saw already some preprints or even papers on this is now, okay, we have in one spot, we have a cytokine profile, but now the cytokine profile may not only act on the nearest neighbors of cells that are sitting there and express the cognate receptor for the cytokine, but it also has a certain sort of, how do you call this? Not half-life, but half distance. So effective distance, the cytokine can basically travel in the tissue, right? So you can now, for every spot where you have a certain signature of a cytokine, you can basically draw a circle where the radius basically gives you some information on how fast the cytokine can travel and where it still has an effect that you can also then check for sort of downstream expression for this signaling pathway that it triggers, right? And there it's also complexity is exploding, right? Because you have all sort of combinations of cytokines, ligands, receptors, downstream signaling, and then you have to sort of run an optimization for every range, how much it goes, and then until you sort of, until it converges, and for every cytokine, then you have a distance. And so there's a lot of data that I see out there, but it's the way it's analyzed is usually always, okay, we have this one molecule that we found in the single cell data, and see it sort of, it co-localizes with our cell that we think makes an effect. So we think the effect is mediated by this molecule that is always very close to it. But again, this is an oversimplification of biology. So yeah, writing down this, you have a source cell, and then you have different layers of cells that can sort of respond to it. But then you also have a property or a feature of every cytokine to travel a certain distance while still not losing potency or being taken up by a macrophage, for instance, and digested. This is also something I think we have to find a better formalism to it. So, I mean, the answer to all of this is, of course, partial differential equations, but then also, again, implementing these optimization routines I just described to estimate, let's say, these effective distances of cytokines. I think this is one of the next steps. Yeah.
Well, if the transport is diffusive, at least you have a concept of a diffusion time and a diffusion length. But if you're, say, in the lung where you have ciliary movement, or in blood, or even in lymph where you have active transport, then it's not obvious that the diffusion length and the diffusion time are the meaningful things. I remember early on in this series, we had somebody who was studying movement of cells in the lymphatic system, and even though there's no pumps, active pumps, at least in human lymph, there are in birds, but not in humans. The rate of transport is actually much faster. Cells move a lot faster in the lymphatic system than you'd expect if you didn't assume there was some kind of active pumping. And so you can get an estimate of diffusion length and lifetimes, for example, and length and time. But that may or may not always be right. Probably tissue local. If you don't have a lot of flow, then it's a pretty good estimate. But otherwise, those are tricky. So I think those are key things. The other thing I was going to bring up, and maybe this is as good a slide as any, when you talk specifically about FAIR, and of course that your award was partly from biomodels, was because you published so clearly, and that's one of the big criteria for that award, is the publication has to be reproducible. And actually the slide you just showed actually was one we talk about, which is, we have pretty good standards now for describing the network and its dynamics. You can use SBML, and you can even describe some of the experiments, computational experiments you do with the setML, although it's not able to do everything. But here you have the diagram, and then you have a set of figures that are derived from those experiments, and something that we do not have good standards for, we could maybe talk about just for a minute, would be, my vision would be, you publish, quote, raw data, and then you publish a set of all the transformations you used to create the diagram. But those transformations, somebody might have used R, somebody might have used MATLAB, somebody might have used Excel. It's not so easy to standardize how you actually go from the data that are generated by your simulation to the diagram itself. And I wondered if that's something you've thought about how you present the results of your simulations, and how you might think about ways of describing how you transformed the simulation experiments into the images that you present in your paper, because that's, I think, actually a critical part of reproducibility if you're talking about a paper, for example.
Yeah. So very good question. So this is exactly what we did in the repository here. So there you find actually all the code that has been used, starting from the raw data, downloading the raw data from the GEO, processing it, setting up the model, calibrating the model, and producing the figures. So I urge my people sort of to document their code very well, but then rather than providing, what's also nice, and what we did in this other story with the red cell cancer that I described, so this is a molecular systems biology paper where we also developed some sort of web interface where people can basically just plug and play and simulate the model. But of course, there they don't really understand, and they don't sort of learn. So of course, ideally, you have both. You have something that is very intuitive and accessible to even non-experts. But what we are aiming here, so one of the rules in my group is that basically we document and upload all the code that is required to run from the raw data until the figures. But then one person is sort of setting this up, and then we ask another independent person from the lab to sort of reproduce it. So this is sort of the first feedback prior to sort of publication before we share it. But then still, we make mistakes, right? And not intended, of course, but people will struggle to reproduce everything. So even for the award, there were people from the judges who were coming back to me and said, we had some problem reproducing figure 2. And then I looked back into the SBML files, and I saw that they sort of confused, I think, 2 parameters or something. So it was not even my mistake. It was sort of their mistake, or maybe I didn't explain it well or something. So it could be solved. All I'm saying is these mistakes happen, and since this is not appreciated or not, there's no incentives, my first impression is, okay, now I feel bad, right? It seems like, okay, I made a mistake, even though it will improve the product, right? So it will help people to better reproduce, but yeah, it's just the incentive right now I have to provide. So for me, it's very important that we provide all of this. We are not perfect at this. I'm sure there's better ways to do it. But this is sort of we learn as we go. So again, I want to provide all the information that people need from raw data to the figures. We do very little post, editing in Illustrator or something. So most of the figures will actually look exactly as you see them in the paper, except for, so in my first papers, I also did these kind of diagrams with LaTeX and ticks. This I don't do right now, but in the others, we also have the SBML sort of standardized versions of the signaling pathways, at least. And also we do these illustrations here, we do them ourselves. So this is also some sort of creative incentive that we have, because I don't like if all the schemes always look the same with this biorender provider. So we do these drawings ourselves. But yeah, so this is my sort of approach, and I will always advocate and encourage people to do it. Because actually also I have a slide, a backup slide, if I can say this. We had a lot of trouble for the IBD story to find human data. So I think I sent out about 17 requests for people to share data where they wrote in the paper data available upon request. And I got a single response. And I got a response there, even though I put in CC all the because I'm a nobody. So I put in CC all the big shots from my institute. Only 1 out of 16 replied and said, yeah, of course, we are happy to share. What do you want? And I said, it doesn't really matter. Raw data, process data, I can work with anything you send me. And then I never heard back again from them. So, but instead of pointing fingers, I mean, there's no point, right? I want to say that at least there was this one microarray data from 15 years ago that was very handy and then proved to be the out of sample validation that we used. So yeah, I would encourage people really to share if they generate novel data, and there is no, you know, patent violated. I really encourage people to share also the data, because this will make it for us also easier to share code that is based on this sort of data. So yeah.
Well, I'm very impressed that you publish all the workflows for generating the figures, because it's a lot of work to pull those together and put them in a publishable form. And it's rare for somebody to do what you're doing. You deserve the kudos for doing that. It's not an easy thing.
Yeah. Jim, I want to comment that, yeah, in the past few years, I started doing that too. I try to provide the codes generating every single figure panels, because sometimes we find out we couldn't reproduce our figure ourselves if we don't really do this.
Yeah, exactly. Exactly. Also, it helps me really, you know, if you have progress meetings or stuff and you want to make things nice, you don't have to start from scratch, but you have a piece of running code that everyone can understand. So you just send it to people and say, okay, help yourself, or adapt it to your needs, or you just rerun it yourself and have it ready. So I think it will, in the future, it will make our lives a lot easier, but it takes some initial, you know, effort to overcome the hurdle. And also, I must say that, you know, I'm profiting from many people who already invested in this. So all the tidyverse pack, so in R now, all the tidyverse packages, right, and the ggpupper stuff that helps us to make things aesthetically also pleasing, right? So once we have the numerics correct, I mean, to make it into an aesthetically appealing figure and to have some standards for legends and corporate, this is also something. So before starting this project, all the people who were spearheading the work sat together and agreed on some sort of corporate identity, how we want the plots to look like, what kind of color scheme we are using. It sounds trivial, but I think, again, this is the first paper that's coming out from the group. And I think people will recognize this, right? I mean, if you have the freedom from the editor to do these kind of small aesthetics and have it consistent, then that's something, you know, once we publish again, people will realize, this looks also in a way similar and reminds me of some previous work. Maybe they do the connection, and they get the feeling that people actually think about this. So yeah, raising awareness, I think, is the first step to make a change.
I mean, it would be nice if there were better tools for defining these workflows and making them available. In other words,
yeah,
you're doing it, but you don't have, there aren't a lot of tools that will help you do this, perhaps, or maybe some. The other end of it, where I wonder about annotation, is when you have, say, a cell type or a gene name or a rate constant, one would like to tag that with an ontological reference. This is pstat 5, and then colon obo colon blah 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 from mesh or Go and so on. When you build your models, do you do that by hand, or are there tools that are built into the modeling environments you use that help you do that kind of annotation?
Historically, I do this by hand because I really started with this bottom-up modeling. So the first paper we published was not even on the JAK-STAT signaling, but was just on STAT5, A and B. So there we knew what we were talking about, and the genes in mice and different species. And then we looked at JAK-STAT, and then we also added MAP kinase and AKT, but all still downstream of the EPO receptor. So yeah, that's why we never sort of took the top-down network annotation topology sort of approach, which doesn't mean that. So I'm not I'm not advocating that one way is superior to the other. I'm just, as I said, I was never used to starting top down and having a network where half of the components I don't know and I first need this reference. But only it was just today that I stumbled about an opinion piece from cell host microbes, a mini review about drug microbe interactions. And there, the authors, I think the last author was Lisa Meyer from Tübingen. They also advocated to have also some sort of drug ID and then some sort of reference IDs for different microbial species, and then also to sort of link them in a more graph kind of approach. So yeah, I'm a big fan of this. So the more we standardize, the more we avoid confusion and mistakes. So I would be a big fan of having one big database where every molecule with every post-translational modification even gets a unique identifier. This I would love, but I'm not aware of this. So as you said, for some sort of omics layers, there are these. For instance, I also know in the sort of lipidome universe, lipids can also have all these very sophisticated cross-links, and they're already managed, as far as I understand, to really reference each of these species with a unique identifier. But again, I'm not aware that this exists for proteins or other molecular layers.
Well, I certainly find, again, at higher scales of organization, cell and tissue scale, many of the core concepts are not given. There are no well-defined term definitions for those. You go into bioportals or the European equivalent, you look up a concept. There is no definition within the ontological universe yet. At the molecular level, it's better, although, as you say, there's still a lot of missing information. But even relatively simple concepts like, say again, your T cell synapsing, you'll find that a lot of the key ideas are missing. And so I'm not trying to be critical, but maybe we need to have you come back and talk specifically about model sharing and the issues you've faced and how you've approached it, because I think that could be in itself worth a presentation. In addition to the great science you've presented today. Are there any we've kept you a long time. Is there any final request for our speaker today from the audience? Well, then I want to thank you very much for coming and giving a very stimulating talk and for being willing to stay late. I know it's late there, so we appreciate the time that you're willing to put up with the time difference as well. Zoom may abolish distance, but it doesn't abolish time zones.